Hi, everyone. Thank you for being here. And thank you for the organizers. I'm Motasem ElGamel, a graduate student in the group of Andrew Mugler at the University of Pittsburgh. And what I want to talk to you about today is our recent work on population dynamics and universal statistics of tumor-inhabiting bacteria. So we all know that our body is full of bacteria. We have bacteria in our gut, there's bacteria in the skin, and these different areas have their own different microbiome and unique composition. And these bacteria, they interact with us, we interact with them when we affect them. So understanding them is essential problem to our health, for example. So no different from that are tumors. So tumors that we get in our body were recently found to have their own microbiome. So multiple species of bacteria were found to inhabit the tumor environment. So they either live in the vicinity of the tumor environment or inside tumor cells itself. And what you see on the right here are different tumor types. In blue are the tumor cells, and in red are bacteria cells. So the question we're going to be trying to answer here is what governs the microbial growth in this environment, right? So to understand this problem, our collaborators, Amir Mitchell and Sir Consign at the University of Massachusetts Medical School, they designed an experiment where they try to probe this environment and try to probe the interaction in such unique environment. So the experiment goes as follows. First, they prepare genetically identical barcoded E. coli strains. So these are exactly the same. They're isogenic bacteria, but you go to part of the DNA and you insert a barcode. And by doing this and you changing the barcode, you're able to get multiple different strains that are exactly the same. So here it's a barcoded library of 39,000 strains that are all the same. You can think of them having different colors. The same time, they have tumor cells, and then you get the tumor cells and you inject them under the skin of mice. And then you wait for a few days, and then a tumor will form in this area. So then you take the bacterial sample, and then you take the mouse, and you inject the bacteria directly into the tumor. And then you wait for a few days, a day, 3 or 7 in this case, and you collect and sequence the DNA, and from that you're able to get distribution of the abundances of these different isogenic subpopulations. So jumping into the results. So this is the injected sample. What you see on the y-axis are frequencies of the different barcodes. On the x-axis is just the label of the barcode index. So barcode 1, 2, 3, the numerical value doesn't indicate something specific. And the frequency means the relative abundance. So how abundant this barcode is in the full population. So then you do the injection, and this is the sample you collect. So you can compare the injected sample in black to the collected sample from the tumor in red. And you see a lot of noise in this statistics. Then you can compare to the normal growth in the lab environment and just an in vitro growth. And you repeat the same experiment, and you see a much nicer curve that's very different from the growth that we see in the tumor. So then I can change the way I look at the statistics here by doing a re-ranking or plotting a frequency rank distribution. And the frequency rank distribution, by re-ranking, I mean I'm forgetting now about the identity of the individual barcodes, and I'm taking the barcode that has the largest frequency, giving it rank 1, the second largest frequency, rank 2, and so on. And when doing this, we find that in the in vitro case, really the growth dynamics did not change the relative abundance of the barcodes. But doing this to the tumor data, we find a different curve. So we find this parallel, it's now more skewed. You can then repeat the experiment, change the collection time. You always keep getting the same parallel. So these are data from collection days after, like day after injection, 3 days after injection, 7 days. And we always get the same pattern. And this pattern is known as Zipf's law. It's the frequency goes as 1 over the rank. So The puzzle here is figuring out the dynamics that give rise to these statistics. And the dynamics in the in vitro case can be very simply explained by a birth process with demographic noise. What this means is just the noise that we see in the statistics is due to the randomness in the division events of the individual bacteria. But the dynamics in the tumor is not known to us. So this will be the task here. So what dynamics that give rise to these observed statistics? And in this case, we have two spaces that are interacting with each other here. So there's the tumor environment and there's the structure of the population. The environment, there's the fluctuations, nutrients, there's the immune system also. For the population structure, I mean the composition, interaction across these different barcodes, and so on. And we don't have any information about the environment, so it's completely unknown to us. It's not accessible, but we have only partial information about the structure of the populations. That statistics follows this Zipf's law. So we start from there. We start very abstractly and ask the question: what is the minimal criteria to give rise to Zipf's law in any dynamical system? Right? And we identify a first criteria is that you need to have no attractor in the dynamics. And this is because if you have a potential that has a minimum, that will translate to in the distribution of the population sizes to a peak distribution. But if you have a flat potential, this will give you like a scale free. So you wouldn't have the peak distribution. And the second criteria is you need to have strong fluctuations, specifically multiplicative noise. This to give you the skewness in the distribution. So you turn this flat distribution to a parallel. So these are two minimal criteria to obtain these type of statistics. So we take these criteria now and we apply them to models of population dynamics. So classical models of population dynamics, a famous example is the generalized Lotka-Volterra model. And the generalized Lotka-Volterra model, you have the N is the abundance of the species I, so Ni. And its dynamics just you have a growth rate and you have interaction across between these different species. Simple example is the logistic growth with just one species. But in this model, you see we'll have always an attractor around some carrying capacity. So it doesn't satisfy the criteria of the no attractor that we identified before. A second class of model is a consumer resource model. And the consumer resource model, we don't have the interactions explicitly modeled, but we have them mediated through the nutrient consumption. So here there's the Sj, which is now are nutrients, and their dynamics is just whatever is being consumed by the species. Translates here to just the depletion of nutrients. An example of this is the Monod model, just one species and one nutrient. And this model doesn't admit an attractor. So we choose this as a candidate for this type of interaction because it doesn't have this attractor. So it satisfies one of the criteria of Zipf's law. And I now take this model and I add to it the second thing, which is the noise. So to go through what these equations mean again, we have the first part is the deterministic growth rate, which is just consuming the nutrient S. We have one nutrient here in the system. And the second equation is that all the species are consuming this nutrient S here. And in addition to that, you have, because this is an open tumor environment, you have flow of nutrients in and out of the system given by this loss diffusion term. And around this deterministic dynamics, we have this noise at the growth rate, the sigma A to I term. So the picture here is that you have the different barcodes and they all have access to the same nutrient and they are consuming it. So let's simulate this model and see how it performs. So the dashed line here is the Zipf's law, the statistics we are looking for. And we see that it's starting to look like a little bit Zipfian. But then you wait for a few more generations, you're losing many of the species. You wait for more time, you're losing more, and you wait and so on. And eventually you get everyone is going extinct. So there is a component here that is happening in the tumor environment that we are not taking into account. So to understand better what this is, we go back to the data and we take a hint from the data. And to do that, we look at the number of surviving barcodes as a function of time. So at day 0, the time of injection, we have 39,000 barcodes. All the subpopulations are there. Then by the first time of collection, by day 1, this dropped to less than 10,000. So we have like 8,000. So this hints at an initial bottleneck that exists in the system. So the population has to go through a bottleneck before invading or like getting established on this in the tumor. So we now want to model this bottleneck, and we model it as so each bacterium will have a specific probability Q to make it through to the tumor. So either live and make it and establish itself, or die. And the probability that a barcode survives is the probability that all of these bacteria, or at least one of these member bacteria of this barcode, will make it. So it's 1 minus the probability that it goes extinct, which is itself is the probability of all of them failing, because they're independent events, so the probability of failure is to the number of cells that this barcode had. And so this would be 1 minus Q raised to N0 being the number of cells in the inoculum times Fi, the frequency of this barcode in the inoculum, how represented it is. I can now look for a quantity that allows me to calculate this Q from data. And this is, we can find this through calculating the average number of surviving barcode B, which is just given by the sum of these PI terms over all the barcodes in the population. So now all these terms, we have access to them from data. So we can accept Q, so we can use the data to estimate Q. And we get an estimate from Q. We find that the probability that one cell will make it to the tumor is very small. That's 0.3%. So, and what this means is that with a probability of 0.3%, the probability of no cells in a barcode survive, so this barcode goes extinct, is 77%. The probability of one cell in a barcode survives is 20%, and 2 cells are like 2.6%. It keeps decreasing. So what that means is that the majority of the barcodes go extinct, but the ones that make it to the tumor just make it with one cell maximum, or 1 or 2 cells maximum. So the picture really after injection, we think that the bacteria will establish itself looking like this, very separated. And when they grow mainly surrounded by members of their own barcodes. So you don't have a well-mixed environment like we were modeling before, but you have this like spatial structure that exists. So now we go on and ask, how do we model this spatial structure, or like include this type of picture in the model? And we add very simple term, which have since you are surrounded by members of your own color, that would mean that you are limited in your growth by competition with yourself. So we added a growth limitation term given by this Ni term here in the denominator. So you cannot grow too much because you are competing with all the other barcodes for the nutrient S, but are also competing with yourself. So we now take this model, and I can simplify it further in the limit of large population limit, and that the nutrients at steady state, and I can write this as an effective Langevin model with just a deterministic term and a noise term, and then use a Fokker-Planck equation to solve for the statistics. And this is what we find. So you can compare to the Zipf's law that we're looking for, the dashed line here, the theory and the simulations, and we find very good agreement. And what's surprising here is that extinction that we saw before is prevented by this local growth limitation. So it's acting like it's making a more equitable access to nutrients, but by limiting growth of very large subpopulations or very large barcodes. So now I can take every experimental curve from these. I can estimate a bottleneck from it, and then I can feed it to the model in the simulation. And you can see here the comparison between the experiment and the simulation for the statistics here for these different days. Okay, so now that we got the statistics, we can now ask about a different quantity that we can verify this theory with, and this would be comparing it to the actual population dynamics. But yeah, before that, I would just want to tell you, so I now modified my criteria and added you need to have this third criteria of an extinction prevention that we found that it's needed to prevent, like to give rise to the statistics. So now the population dynamics is, so you can see that the data here, the circles here are the CFU data, the conforming units data, and our model is given by these lines. And you can see the blue ones are the CFU, and the ones below, the pink ones are the surviving barcodes. And this here shows that it shows an initial decrease in the total population, but then we think that this initial decrease has to happen due to the bottleneck, but then everyone will, like the whole population will go up. And we can now look at another independent source. So there is another paper that now shows after 1.5 hours of infection, you have this initial decrease from the initial injected population. And then you get, so this hinting at also the existence of a bottleneck, and then you get this final increase like we see. So this is a confirmation of the type of dynamics we think are happening in this environment. So I started with this picture that I have very partial information about the system and with an environment that's completely unknown. But now we were able to like develop a theory that can inform us better about what type of interaction can be happening here. So we now have a better understanding of the environment. So it has fluctuation, competition for nutrients, infection bottleneck, and for the structure of the population. We think that we hypothesize that there's the niche specific growth slowdown that has to exist in this structured environment. So that I would like to end. I'd like to acknowledge my advisor, Andrew Mugler, our collaborators, all the Mugler group members. We have a preprint if you're interested in checking the work. Yeah, thank you so much for listening. I'm happy to take any questions.
Yeah, that was super fascinating. So when you showed the Zipf's law, I was wondering if like there's a whole like theory in ecology about species abundance curves, which I don't know very well, but I think like Zipf's law is one of the observations there, and a lot of modeling for how that arises in natural ecological systems. So I guess how much of what you see do you think is specific to the bacteria in the tumor environment, and how much of this is like ecology, as a broad theme?
I think it's not specific necessarily to the tumor environment. So the criteria we identified is very general, can be applied to any system. We just applied them in this context. So it can be shared across many different ecological systems. Yeah.
I was curious about how you dimension the model. Like, are you able to be very accurate on the parameters that you put there?
How you dimension the model? You mean how do we know the parameters that we choose?
Yeah.
So yeah, so it's a good question. So the parameters that we choose, we chose the growth rate to be consistent with like the growth rate of bacteria in very rich environments. So we assumed division time every like 30 minutes. And we similarly for the other parameters, we estimated just biologically what would be a diffusion of a nutrient, what should be the diffusion constant of the nutrient in general, like in any environment. So these we fixed them not specifically by knowing anything specific about the environment in the tumor, but just from like what is biologically sound. So this is how we chose the parameters here.
So I was sort of curious, I couldn't quite tell from the setup. Do you guys have sort of replication data either across mice or over time that you can sort of test aspects of your theory about the sort of single cell bottlenecks with?
Replication data across mice or time?
Yeah, I guess I'm imagining if everything's founded by a single cell, if you do the same inoculation in 2 different mice, those lineage sizes should be totally uncorrelated, for example.
Yeah, so these are different. So the data here is from, so every curve is from a single mice at a specific day. So these are different mice, same inoculation. And they are uncorrelated. Because these are barcode depicted, so you can tell which barcode is showing high frequency here, and they are uncorrelated. They are correlated in because they have two tumors. So there's a tumor on the right and tumor on the left. But I think it wasn't also conclusive if it's always the same barcode that will make it. But yeah, across mice, it's not correlated.
Thank you very much.
Thank you.